Okay, we start with the topic that is antiviral drugs, and antiviral drugs comes under the antimicrobials, and it's coming to the why these drugs actually we use for the treatment of viral infections. So coming to the virus, so virus is an infectious particle, okay, in microorganism, okay, and it infects the host cell. And the difference, if you see that the difference between the bacteria and the virus particle, if you see that, well, bacteria, it can replicate on its own. It has its own mechanism to replicate on its own. Whereas if you see the virus, it cannot replicate on its own. It has to use the host mechanism for its replication. So, and virus can infect any type. Okay, it can infect humans, or it can infect animals, or it can infect bacteria also sometimes, or it can infect even plants also. And so virus is a small infectious agent which causes the disease in the especially humans, but it can infect others also. And if you see the viral structure, okay, so this is the basic viral structure of any virus. And here if you see that you have a layer, okay, and on the outer layer you can see that there is a Gp120 protein. So this is the most important glycoprotein for the virus, and these glycoprotein only recognize the host cell surface proteins. And this that is glycoprotein 120. It is very important protein for the virus. You can see that on the surface, all these are all glycoprotein 120, and on the surface, viral particle surface, you have all these glycoproteins. So this is especially glycoprotein 120, and it is very important for virus because this protein, it detects the host cell and to attach to the host cell. Without this protein, the DNA, now that is virus cannot attach to the host cell and can cause the infection. So this is one. And you have its inside, if you see that you have its a genome, either it is an RNA or it is a DNA, and then you have the other enzymes, okay, and like integrase you can have, or you can have reverse transcriptase, or you can have a protease enzyme. So inside you have all the materials for the virus, and then it is covered by a capsid. You can see that blue cloud, okay, it is covered by capsid, okay. Why? So inside the capsid, what you have is its viral genome, okay, and then the enzymes which are required for the replication. Then you have the matrix here, okay, and next one you have the lipid membrane, outer lipid membrane, and then so this is the basic the structure of your virus particle. So virus particle, you will have the outer lipid membrane and capsid, and inside the capsid you will have the its viral genome and their enzymes. This is virus, the basic structure of virus, how it looks, main components of virus. So if you see the main components, you have the one enzyme is protease enzyme. You remember this protease enzyme, and then you have the inside genome, that is RNA, or it can be DNA, and then again you have the reverse transcriptase enzyme, reverse this, and another one you have the integrase enzyme. So these are the enzymes which are present in the, okay, with the virus. And one more thing is that it is very important in the virus, it is the Gp120 protein. So this is also glycoprotein, which is a docking protein, means this protein recognizes the cell surface of infected cell. There is a whatever the humans or bacteria or plants or any other kind of animals. And next, if you see, that is a virus particle. So the main components of virus particle, you can call it as a virion also, and genetic material, that is one component, and a protein coat, we have seen the capsid, and then envelope, that is lipid membrane. So these are the three things which are present. So you can see in the slide, genome, genetic material, and some enzymes, and a capsid, and lipid membrane. Okay, lipid membrane. So these are three components present. Next one is that viral life cycle. Because why we are discussing viral life cycle here, it is that, so already you might have studied basic things about the virus. Here viral life cycle, because the treatment of the viral diseases majorly, it, so it depends upon the drugs what we use, it depends upon the virus life cycle. So that's why we are going to little detail, not full detail, and brief we are going to see what the viral life cycle. So the first step is, it is attaches the host cell. So as I told you, the attachment happens to the glycoprotein recognize the cell, and then virus goes and binds to the, attaches to the your host cell, whatever in that is, whichever the cell it is infecting. Then once if it attaches and it diffuses into the cell, after attaching, the virus particle diffuses into the cell, and after diffusing, and it uncovers itself, and then releases its genetic material and then enzymes. And once the genetic material is released into the host cell, okay, then it starts replicating, synthesis of it, proteins, enzymes, all these things will be synthesized. And then once the whatever the component, whatever all the components are required for the virus, okay, they will be synthesized. And once they are synthesized, all those components are assembled together, and then it will be made into a virus particle. Then it comes out of the, once virus particle is made, okay, and assembled, then it comes out of the cell, infected cell, and to further infect the other cells. Okay, so it will infect, that is we call it as a release phase. Okay, this is the briefly the life cycle of virus. So the viral life cycle, it starts with the attachment of virus to the host cell, that is whether it is a human cell or whether it is a, so in humans maybe it may interact, that is bind with the blood cells or other cells it can bind. And then once it binds and it diffuses into the cell, once it diffuses into the cell, then it releases its material, okay, that is enzymes and genetic material. Then once the enzymes and genetic material released into the host cell, replication will start, and synthesis of components will take place. All these things are made, once all these things are made ready, and all these things are made ready, then they'll be assembled into the virus. Once a viral particle is assembled, then it will be released out of the cell, and these released virus particle again further infect the other healthy cells. So next, as I told you, the virus genetic material, so you can call the basic broadly classify the virus into two types. One will be of either it is a DNA virus or RNA virus. So DNA virus, you'll have the DNA deoxyribonucleated, RNA ribonucleic acids. Okay, so this is the basic difference. And the DNA type virus and RNA type virus, basically these are the broad classification based on the genome. And the DNA so can be of double stranded DNA or single strand DNA. And whereas RNA case, it will be general, it will be of a single strand RNA. So that is the class how you classify virus. So the virus can be a DNA type of virus or RNA type of virus. So DNA type of virus, you'll have the DNA, and RNA type of virus, you'll have the RNA. In DNA type of virus, you will have double stranded DNA or single strand DNA. Whereas in RNA type of virus, you'll have the only single strand. So this is the life cycle. You can see in the slide. First, the virus particle, what it does is that through Gp120 protein, it recognizes the cell surface proteins of a host cell and binds to the host cell. And then next step is that diffusion into the cell. And then once if it is diffused, so all its materials will be released into the host cell, and then replication takes place. Okay, and once the replication and synthesis takes place, again the virus will be assembled. Okay, virus, and then again further it comes release phase, it comes out of the host cell, mature viral particle, and this further goes and infects other healthy cells. This is life cycle. And the infections which they cause various infections, they can cause viral infections. Okay, so like you have, these are all the infections virus can cause. Okay, so we have millions of virus, but we don't know many of the viruses till now. I think only about 5,000 virus, but completely about 5,000 virus completely it is studied. So these virus, okay, so if these are the infections, they can cause the virus. So meningitis it can cause, okay, and then that is we call encephalitis, like you have measles and rabies and pharyngitis, adenovirus, and cardiovascular diseases, okay, and hepatitis, okay, A, B, C. You have skin infections. The virus also can cause the skin virus, skin infections. So that is human herpesvirus, smallpox, okay, rubella, measles. All this comes under the skin infections. And then the virus can cause the eye infections. That is herpes simplex virus, HSV virus, adenovirus. You have then pneumonia. So influenza virus. So we know that they can cause the pneumonia and respiratory, okay, single virus, and then adenovirus it can cause the now we recently we are dealing with that SARS or coronavirus that is also causes the that is that is pneumonia. The presently what we are dealing with, that is SARS coronavirus and myelitis. Then you have the poliovirus, okay, and gastroenteritis, okay. So adenovirus, rotavirus. So these are all they can cause gastroenteritis and pancreatic infections, okay. And then sexually transmitted diseases. So majorly you have the HIV, okay, herpes, okay, that is simplex type 2. So these virals can cause of any of the organ, they can cause the infection, they can have the infection, okay. And coming to the treatment, okay, so the antiviral drugs, okay, these are the class of medications used for the treating of viral infection. So the treatment can be targeted here, it is not generalized targeting. So here compared, that is if you see that viral drug, so the viral drug can be specific to a particular infection or a broader, that is broad spectrum, but generally we don't use the broad spectrum. So here the treatment is for a particular type of virus, and the targeting here we have, it is the virus treatment targeting. So there are approaches how we treat the viral infections. So the first one is that immune system stimulation. It's our own immune system we stimulate so that our immune system can fight with the virus, okay, and eliminate the virus from our body. So especially alpha interferons, if you stimulate them, so they can fight against the virus. Next one is antibodies. Our own antibodies, if you see, they are not antibodies, okay. So you can stimulate the production of antibodies, and these antibodies in turn can eliminate the virus particle from our body. So the antibodies can be of polyclonal or monoclonal. So polyclonal are not specific, whereas monoclonal antibodies are specific to particular antigen. So here in our case, here it is virus. So to a particular virus, you can synthesize a monoclonal antibody, okay, and this, once you give the monoclonal antibody, so this monoclonal antibody can fight with the virus. And one more thing here, you have to see that virus antiviral drugs, so they don't kill the virus. You should understand, they don't kill the virus, they only stop the replication of virus. And virus without replication, it cannot stay much more time in a host. So you have seen that bactericidal and bacteriostatic. Bactericidal, if you see that they kill the microorganism, and others on cidal, it is that they inhibit the growth of the microorganism. Same way here also, antiviral drugs, basically they do not kill the virus, they only inhibit the replication process they inhibit. So here the targeting of viral for the treatment of viral infection, so you can have various strategies. So one strategy is that stimulation of our own immune system. So the immune system, especially the of interferons, and then next one is that antibodies. So antibodies, that is our own antibodies, are of polyclonal. That means they're not specific to the antigen. So they can go and bind with any kind of antigen. they're not specific. And if then you can have the monoclonal antibodies. Monoclonal antibodies means they are specific to the antigen. Okay, a particular way. If it is meant for the HIV virus, and it goes and binds to the HIV virus and eliminates the HIV virus, fights with the HIV, sorry, HIV virus. So that is one strategy where you can have the that is treatment of that is viral disease infections. Next one, our second strategy is that now we have studied the life cycle of them. We have in the previous slide, we have before 2 slides, we have seen that what is the life cycle of virus particle is involved. So based on the life cycle stages, you can have the treatment. Okay, you can target each step of the life cycle, and you can prevent or stop the replication of the virus. So first one is that, okay, approaches, that is before cell entry. Before cell entry, we know that the virus particle, first it has to recognize the host cell, and then once if it recognizes the host cell surface protein, then it enters into the cell. So once if it enters in cell, then replication starts. So before that is happening, before the virus particle is entering, then you can target there. So, and you can stop the replication of that is virus. So under that, one such condition is that one is that entry inhibitors and uncoating inhibitors. Okay, these are the 2 classes of the drugs you can use before the virus enters into the cell. Okay, before virus, or before binding into the cell, or before diffusing the virus into the cell. These 2 classes of drugs you can use, entry inhibitors and uncoating inhibitors. Okay, so but still now we do not have the entry inhibitors, but we have the uncoating inhibitor. Uncoating inhibitors, we are there. We in the market, they are there, uncoating inhibitors. Next one is that during the viral synthesis, once, okay, if it enters into the cell, next step is that what are the components are there, what are the components are required to assemble the virus particle. So those will be synthesized. DNA will be replicated, or RNA will be replicated, or the enzymes which are required for the virus, those the like integrase or protease or reverse transcriptase, all this will be synthesized in the body. So our host cell. So that is during viral synthesis. So you can, here during the viral synthesis, you have various targets. So of that particular one of the target is reverse transcription. So that is especially present in the HIV virus. Okay, that is a retrovirus. We call it as a reverse transcription because you have the presence of enzyme called as a reverse transcriptase. HIV virus contains the enzyme called as reverse transcriptase. So, and that is that you can do it. You can target the enzyme reverse transcriptase, or you can target the integrase, that is another enzyme which helps the virus for the viral synthesis. And the next one is the transcription. Okay, that is mRNA will be transcribed, okay, into proteins which are required, and translation or antisense. Okay, this translation or antisense we are not going to discuss, okay, since it is not your that is beyond your syllabus. Okay, so these are the first target for our that is based on the life cycle is before cell entry. Okay, that is entry inhibitors or uncoating inhibitors. So and then next one is that during the next stage is that during viral synthesis. So under viral synthesis you can target reverse transcription, and then integrase, and or that is integrase and an enzyme again. Okay, that is, and transcription and translation. That is, these are the things you can do. And then protein processing. Once if it is synthesized, proteins are synthesized, okay, for the enzymes, and they have to be processed. During the processing those proteins, there is an enzyme called as a protease enzyme. So this protease enzyme normally in general sense if we take, okay, so it converts, okay, immature protein into a mature protein. Okay, protease enzyme. So by slicing the protein, immature protein will be converted into mature protein. Next one is the assembly phase. Mostly in assembly phase means all the components are synthesized, and once they are synthesized, and they have to be combined, and then a virus particle will be made. Okay, so that is assembly phase. And release phase. Once the virus particle is assembled, by including all the components, then the virus particle is released out of the cell, infected cell. So that is release phase. So you can target, these are the approaches we have for the antiviral, that is antiviral drugs for the treatment of viral infections. So what are the strategy? First strategy is that, so stimulate the immune system. And second strategy is that you target the virus based on its life cycle. So the life cycle wise, if you see, before cell entry, okay, and during the viral synthesis, that is after cell is entering into the diffusing into the cell, virus particle diffusing into the cell. Next one is that once the diffusion is there, synthesis is there, and then making the proteins, immature proteins into mature proteins, that is using the, that is targeting the protease inhibitors, and then assembly, or that is that second step also is over, and third step it is once the synthesis takes place and the synthesized components has to be assembled into a virus particle. During that stage you can use, and then the release phase. Once the virus particle is assembled, then it gets released from the cell, host cell. During that phase also you can target. So these are the targets approaches we have for the treatment of viral infections. So coming to the drugs, okay, so first one is that entry inhibitors. So you don't have in syllabus, you don't have to take it down. So this is the only drug now it is available as entry inhibitor. So this entry inhibitor, now normally what happens is that they are also called as CCR5 receptor antagonist. CCR5 receptor antagonist, and the CCR5 receptor is a co-receptor. CCR5 is a CCR5 receptor is a co-receptor, and this receptor helps the virus particle to migrate, to diffuse into the host cell. Without this CCR5 receptor, virus particle cannot diffuse into the host cell. So here, that is CCR5 means that is chemokine receptor type 5, or we call also, or CD195, and it is generally present on the white blood cells, okay, which is involved in the immune system, and that is white blood cells. And miraviroc, it is the one of the, that is first entry inhibitor. And how it acts is that it is, see that, okay, and you in the pink color, if you see that, that is a virus particle, and then that is on the virus particle, if you see that these are the glycoproteins. The glycoproteins I have explained in the structure of virus. So these are glycoprotein Gp120. Okay, so this protein, first what it does is that it attaches to the CD4, okay, complementary, okay, CD4 cells on the cells. And once they attach into the CD4, so now next step you see, so here also, so that is CCR5 is there, CCR5, and once the virus particle attaches to the CD4 complex, and then this binding brings certain configurational changes, conformational changes in the CCR5, and once the conformational changes comes, then the virus particle binds to, you can see that it is now, it is binding to the CCR5. This is CCR5, and then once if it binds, then your virus particle diffuses. Okay, so this is how CCR5 receptors helps the virus particle to diffuse into the cell, host cell. So virus, while entering into the, okay, host cell, first it will bind with the CD4, and after binding to the CD4, there will be a conformational change in the CCR5. Once the conformational change is there, then the virus will bind to the CCR5 and then diffuses. So then once if it diffuses, then the virus will get, replication takes place, synthesis, all the synthetic steps will take place, fine, and they'll multiply. So this is now what we are doing, entry inhibitor, that is maravirac. So this is an antagonist of CCR5. So this inhibits the CCR5 receptor, so that virus will not bind to the CCR5, so that it cannot enter into the cell. So this is entry inhibitor. So next class of the drug is that, okay, this is again comes under before, that is before cell entry, that is uncoating inhibitor. Uncoating inhibitor. So now suppose the thing that virus is entered into the or diffused into the cell. Once if it diffused, once if it is diffused, once if it is diffused, then it has to replicate. Okay, so for replication, so what happens is that you have the outer cover of virus. That outer cover has to be removed, and once if outer cover is shredded, okay, outer cover is shredded, so then whatever the genetic material or enzymes, they come out of the virus particle, and then this genetic material and enzyme, they start using the host mechanisms, they start a replication. So that is, we call it as a first. So once if it is enters into the cell, then the genetic material has to be released into the host cell. So releasing of genetic material and enzymes into the host cell, we call it as a uncoating. So without this uncoating, so here virus cannot replicate. So first it has to be uncoated, and genetic material enzymes has to be released into the host cell. So here the previous slides we have seen, that is on, I'll show you once again. So here you can see that. So first is binding to the, okay, CCR5 or CD4, then diffusion. Once diffusion happens, so the genetic material and everything is released into the host cell. This step we call it as a uncoating process. The outer membrane, that lipid membrane will be, or the genetic material will be released into the host cell. So that we call it as a uncoating. So now here the next class of drugs based on the cell cycle, that is, so you have here uncoating inhibitors. So the uncoating inhibitors, so examples you have, or amantadine and rimantadine. So these two structures you can write it down. So I'll give you some time and you can write it down. Amantadine and rimantadine. So these two are uncoating inhibitors. So have you taken down? If you have taken down, any one of you can raise so that I can go to next slide. Okay, you have taken down. So I'll go to the next slide, and the next class of drugs. Okay, so I'll go to the next slide, and next class of the drug, that is uncoating inhibitors. Uncoating inhibitors example I have given. So here, how the uncoating happens. So this is a virus particle. So inside the virus particle, that is the lipid membrane, you have inside there is a capsid will be there, and inside that you have the genetic material inside. That genetic material has to come out of the virus. Okay, so how it comes out of the virus is that, okay, so how this amantadine and rimantadine, they inhibit the release of, that is uncoating. So here you follow my cursor. So now you can see there is a white thing you can call. So that is M2 porins, they are called as. You can see there one, and you can see here another one. Okay, and you have the everywhere on the cell surface. So you can see here one. So the white thing, whatever I have, my cursor is moving, you can see here and here. Okay, here. So everywhere you can see that whichever, these are called as the M2 porins. Through this M2 porins, what happens is once the virus particle, listen clearly to me, okay, carefully. So once the virus particle enters into the host cell after diffusion, so what happens is that, so here through these porins, hydrogens will enter, protons will enter into the virus particle, and protons will enter proton will enter into the virus particle and causes the acidification of the cell, here virus, and causes the rupture of the cell, that is the lipid membrane. And then here what happens, here the whatever the genetic material will be released into the host cell. So here the amantadine and rimantadine, what they do is that they bind to these porins and then prevent the migration of proton from cytoplasm, that is from host cell cytoplasm to the virus particle. Okay, so to release the material, the virus particle has to be acidified. Okay, the acidification of the virus particle happens through these porins, that is migration of protons through these porins called as a M2 porins. M2 porins, that means pores. Through these porins, proton will migrate, okay, from host cell to the viral particle, and once proton is migrated, and it will be acidified. Once if it is acidified, and the genetic material will be released from, it gets ruptured off, and genetic material will be released from the virus to into host cell. Okay, so now I'll stop this meeting. 1 second, be on the thing. Yeah, so I'll stop this meeting now. Again, I'll connect with you in again I'll connect remaining topic to finish. Okay, so be online. Okay, I'll again within 5 minutes I'll connect to you so that we can complete the topic here. Is it okay?
Okay, sir.
Yes, sir.
Yeah.
Yes,
So
sir.
till now, did you follow?
Yes, sir.
Yes. Yes,
Have you
sir.
followed? Have you followed the class, or is there any difficulty to understand the class?
No, sir.
All of you have followed, correct?
Yes, sir.
Okay.
Yes, sir.
Then yeah, within one minute, I'll 5 minutes, I'll come back to you. Okay.